Our next speaker is Dr. Trentacosti. Oh, there's good. There she is. Come on up. Dr. Trentacosti is going to give us the U.S. perspective. She's a senior medical reviewer with the Office of New Drugs, the Center for Drug Evaluation and Research at the FDA. Her current position is acting labeling team leader in the study endpoints and labeling development. It's a sealed group. She advises review divisions in matters of regulatory policy pertaining to the development and validation of study endpoints, including measures of treatment benefit regarding products for gastrointestinal disorders. Thank you very much for being here.
Thank you very much, Brennan. Well, I've been asked to talk about unique problems in multinational studies concerning endpoints. And what I realize in identifying unique problems, I also have to discuss some not-so-unique problems that are pertinent to all clinical trials. So clinical trials are going global. And actually, probably the more accurate statement is that they've been global, but that that's increased over the past decade. In fact, FDA is seeing more and more applications from that includes data from non-domestic sites. For example, in fiscal year 2008, 80% of approved marketed applications for drugs and biologics contain data from foreign clinical trials. During that same time period, over half of all clinical trial subjects and sites were located outside the United States. And this trend is dramatically increasing. So FDA is left with this increase in foreign data and trying to interpret some of this interregional variation in study results that we're observing in the data. And certainly there's multiple factors that can be contributing to this interregional variation, including chance, patient selection, ethnic diversity, unblinding, or differences in study conduct. I'm going to focus on ethnic factors, which my two previous regulatory colleagues did allude to this guidance, which is the International Conference on Harmonization, Guidance on Ethnic Factors in the Acceptability of Foreign Clinical Data, also fondly known as E5. And this guidance was published by FDA in 1998. And this guidance gives an overall regulatory framework on developing drugs in a global perspective and how to evaluate those ethnic factors and how they relate to outcomes, and also ways to minimize duplication of clinical trials in a global development program. So E5 actually defines these ethnic factors as factors that relate to race or larger populations grouped according to common traits and customs. And these factors actually can affect almost any aspect of a drug development program, including the safety, efficacy, dosage, and dose regimen. The impact can vary depending on many factors, including the pharmacological class of the product, indication, and age or gender of the patients. E5 goes a step further and actually classifies these ethnic factors as intrinsic or extrinsic. So this slide here actually differentiates these intrinsic factors from extrinsic factors. And the intrinsic factors are those that really define the population or identify the subpopulation. And they can be genetic or pathophysiologic. Examples include gender, race, genetic polymorphism of a drug, metabolism, comorbidities, severity of the condition. Extrinsic factors are those that are more defined by the patient's culture or the environment in which they live. And examples here would include socioeconomic class, education, language, medical practice, diet. E5 focuses a lot, primarily on the intrinsic factors and less so on the extrinsic factors. So this heterogeneity of data certainly can be very problematic from a regulator's point of view. And if the data is that significantly different, it can actually result in a non-approval or possibly the need to request more data or additional studies, or at the very least maybe labeling. We may need to include some of this information about the ethnic factors in labeling. And as we talked about patient-reported outcome measures, these are very often used as primary endpoints in symptomatic conditions, including functional gastrointestinal disorders. And as subjective measures, PROs can be especially influenced by extrinsic ethnic factors, particularly language, culture, and socioeconomic status. So this is FDA's PRO guidance, and the PRO guidance actually explains how FDA reviews PRO measures that support labeling claims in product development. And my contention is if you follow the principles and measurement properties of the PRO guidance, it actually could help minimize some of these extrinsic factors. So in order to bring up about this point, I'm going to explain some of the key points from the PRO guidance. So one of the first things we look at is whether the actual endpoint or concepts support the targeted claim. And this seems like a very simplistic concept, but actually it's often very problematic. And I'll explain this in an example a little bit later. Once we determine that, the next step is to actually look at the instrument and the measure. And as Brennan nicely pointed out and discussed, we look at content validity, which is one of the most important things we look at. And content validity is essentially the evidence that the instructions, items, and response options are appropriate and comprehensive relative to the intended measurement concept, population, and use. So essentially, is the measure actually a measure of what's supposed to be? And there's two methods that we use to evaluate the content validity, which again, Brennan discussed. One is concept elicitation, and those are the open-ended questions with the target population. We're trying to understand the symptomatology from the patient's point of view using their own terms. Once we've actually captured that data and understand the concepts, we develop our instrument and our items, and then we go back to that same patient population with cognitive debriefings to ensure that the patients understand, interpret the items, and is still comprehensible. FDA also then looks at the content validity of the translated and culturally adapted versions of the measure. Now, certainly it may not be necessary to do the entire concept elicitation and cognitive debriefing is in detailed as in the original instrument. At the very least, however, cognitive debriefing would be necessary. But for a simple concept, maybe like just pain intensity, you could probably stop with cognitive debriefing. But with a more complex concept, such as irritable bowel symptoms, it may be necessary to evaluate that concept through more open-ended concept elicitation interviews with the patient population. We also look at the process used to translate and culturally adapt the instrument. And certainly we look at issues that were observed during this cultural and linguistic translation and how they were adapted into the new measure. Recently I reviewed a measure in which it was noted that the clinical research associate from each country translated the measure. That was the sum total of the translation linguistic process. And that certainly did not give us enough information to determine whether it was adequately translated or not. Then we go on beyond. Once the content validity has been established, we go on to look at the other measurement properties, which are reliability, construct validity, and ability to detect change. Now, certainly we wouldn't expect a sponsor to look at this in every single different translated version of the measure. The original measure, yes. But we would recommend, certainly, that you at least test the measure in a phase 2 trial before you actually implement into a pivotal trial. We look at the data collection method, and it may be useful to use an electronic version in one region and a paper version in another region, which is perfectly acceptable. However, we just want to ensure that those two methods are comparable. We look at the review of the instrument format, instructions, and training. And I have to say, training, I think, is very key. Training both the investigator as well as the patients in how to complete these patient-reported outcome measures. And then we look at the logic and methods used to interpret the clinically meaningfulness of the trial results. So I'm going to give a little example here of the SPARC trial, which is Satraplatin and Prednisone against refractory cancer. And certainly this is not a gastroenterology trial, but I think that the points in this trial can be extrapolated to any multinational trial. So the indication proposed was treatment of cancer with patients with hormone refractory prostate cancer who had failed chemotherapy. So these patients had already had to experience at least one round of chemotherapy and failed it, and they had to have evidence of metastatic disease. And the study design, it was a multinational trial with involving 16 regions, randomized, double-blind, placebo-controlled. And patients were randomized to either Satraplatin and Prednisone or Prednisone and placebo. And this first brings up one interesting point, which is unintentional unblinding. Certainly, these patients have been exposed to a prior chemotherapy. They are well aware, unfortunately, of the serious adverse reactions and toxicities associated with these products. So the patients assigned to Satraplatin and Prednisone would certainly have different adverse reactions than the patients reporting to placebo with Prednisone. So this could lead to unintentional unblinding both from the investigator as well as the patients, and that could, of course, affect the reliability of the data. Now, this is a co-primary endpoint. One of the primary endpoints was progression-free survival, and the other was overall survival. This information was presented at an advisory committee meeting, and the only information at that time available was this progression-free survival data. And this was a composite endpoint that was defined as from time to randomization to first occurrence of any of the following: death from any cause, radiographic progression, skeletal-related events, and symptomatic progression. Then the symptomatic progression was again defined as either worsening performance status, 10% weight loss, clinical events attributable to disease or prostate cancer, or pain progression. So the first question asked the advisory committee meeting, which is the first question we asked, is does this endpoint adequately represent or measure the concept or would support the claim of treatment of prostate cancer patients? And at the end of the day, after looking at the data, the advisory committee meeting actually decided to wait for the overall survival data. But I'm going to focus on this pain progression endpoint first. So pain progression was assessed by a daily diary that patients had to complete every day in which they measured their pain as well as their analgesic use. And the definition of a pain progressor was somebody who actually either increased their pain intensity score or analgesic use compared to baseline. This is an example of the present pain intensity item that was asked of patients. So report average pain intensity over the past 24 hours. 0, none, 1, mild, 2, discomforting, 3, distressing, 4, horrible, and 5, excruciating. So we had significant content validity issues with this item. The sponsor did not provide any significant or satisfactory information to support the content validity either in the original instrument or in the translated versions. And so, of course, there's concern. Do patients actually use this terminology? Is it clinically meaningful in describing their pain? Does a 2 really mean discomforting and a 4 horrible? Is the average of discomforting and horrible really distressing? So there was a lot of concerns about this particular pain intensity item. Now, this is an example of the pain analgesic diary. And this is something we commonly see, and that sponsors will pay a lot of attention in evaluating the content validity of the questions, but not necessarily the format. So the question is this: in this diary, patients are asked every day to actually note, the bottom here is their present pain intensity, and over here they're supposed to note the analgesic, the name of the medication, the dose, the number of tablets they took. This is an elderly patient population, so there was no information provided if patients could actually complete this satisfactorily. Certainly there's a lot of concern about this paper version of the diary in that patients could review their previous responses. So if at day 7 I forgot my previous day 1 to 6 responses, I could just fill in the data. Or day 4, I could see my response for day 1, 2, or 3, which could affect what I put down for day 4. So we had some issues with certainly the content validity of this diary. And then, of course, there's a whole issue of patient burden. Patients had to complete the same diary every single day for months, and which brings up a whole new issue of course, missing data as a result of requesting that from patients. So this is the percentage of patients with pain progression from each region. And if you see over here in the legend, you can see the wide variety of regions that were included in this trial. The only country missing here was Canada, which only actually enrolled 3 patients. And certainly when you have a large variety of regions like this in multiple sites, you can enroll patients more rapidly and complete your study more rapidly. Also, you're having greater exposure to your product, but it also is going to increase the heterogeneity of your instrument and, of course, all the other factors associated with the ethnic factors. And if you look at this, there's certainly a discrepancy here in the percentage of pain progression. Now, remember, these patients did have metastatic disease, so you would expect most of them to have pain progression. So if you look at countries, say here the United States, where almost 40% of patients had pain progression, compared to some of the countries down here, say Spain and Belgium, which less than 20% had pain progression. And what's interesting about this is that although this regional difference was noted for pain progression, there was no regional difference noted for radiographic progression. And certainly, of course, there's a lot of other contributing factors other than just heterogeneity in the ethnic factors that could be contributing to this, but this is certainly something that could be contributing to this. Now, this is the percentage of pain progression attributed to increased analgesic score. Now, based on our review from the United States, our culture, we would assume that most patients would have pain progression based on increased analgesic score as compared to increased pain intensity. Because to be a pain progressor, you had to have increased score for greater than 2 consecutive weeks. So it would be our culture that the physician would increase the analgesic of the patient rather than letting them suffer in pain. But again, we're seeing a lot of variability in this data. So if you look here, for instance, Israel, almost 98% of the patients here did have an increased pain progression related to increased analgesic score. But if you compare that to, say, Germany, where it was much less, 30%. So that's, again, a different discrepancy, which certainly the heterogeneity of the regions could be contributing it. So in summary, based on the E5 guidance, certainly there's intrinsic and extrinsic ethnic factors that could be contributing to heterogeneity of the data. PRO measures are especially probably more influenced, impacted by these extrinsic factors because they are subjective measures. But paying attention to the principles and measurement properties of the PRO guidance can help minimize the heterogeneity of these extrinsic factors, the adequate measurement properties from the original instrument, of course, and then that the culturally or linguistically adapted version are also comparable with that of the original measure, and certainly an adequate language and translation process.